According to the syllabus, this is what you've got to be able to do. Number one, explain what all those terms mean. Allele, variant, basically the same thing. You've got to explain what dominant is, recessive, homozygous, heterozygous, genotype, and phenotype. You've also got to be able to explain something called single gene inheritance, which is something we'll talk about in a few minutes. Also predict the results of single gene crosses. That's linked to the previous objective. Recall that most phenotypic features are the result of multiple genes rather than single gene inheritance. In other words, eye colour, for instance, blood groups, they are not regulated entirely by one single gene. There's more than one involved in making your eyes brown or blue or green or giving you an AB negative blood group and so on. Describe the development of our understanding of genetics. Well, that's kind of going to come through during this whole lesson, really. Just going back in history, this whole genetics subject, I suppose you could say, was started by this guy. He was a monk, Augustinian, like it says there, in what is now the Czech Republic by the name of Gregor Mendel, born in 1822, eventually died in 1884. And as well as his responsibilities at the Abbey, he also taught natural science, so biology, at a local school. While he was at the Abbey, he actually looked after the greenhouses, which turns out to be kind of his claim to fame, because in 1843, he started his release. That's when he kind of published his work, but he'd been doing it for a few years. And he found that the plant had various characteristics where they were either one thing or the other. So, for example, the seed shape was either spherical or wrinkled. There was nothing in between. They were either spherical or they were wrinkled. The seed colour was either yellow or green. No in-betweens, no sort of greeny yellow. It was either yellow or it was green. The flower colour, a reddy purple or white. Now, he did try originally to grow pink flowers, sort of halfway between the two, but he couldn't do it. However many times, he tried crossing the purple, so the dark reddy purple ones with the white ones to try and get an in-between colour, and no matter how many times he tried it, he couldn't do it. That's when he started to realise, obviously. And then he also looked at the pod shapes. They were either pod shaped or actually tight around the peas. The pod colours were either green or yellow. And there's nothing. And then look at the flower position and the stem height, either tall or dwarf, axial flowers or terminal flowers. There was no in betweens. There was no mixtures. So in one of his experiments, just like with the flowers, he made a tall plant with a dwarf one. Now, what he would have expected, obviously, was a medium-sized one, because, well, you would, wouldn't you? You get a tall person, you mate them with a short person, you would expect their children to be somewhere in between, because, mixing. Mixtures. Turns out what he got was that all the plant came out of this cross were all tall. So what he concluded from that, it wasn't just a one-off. He'd done this hundreds of times. He'd done all the probability calculations because old Mr. Mendel was a bit of a maths geek. In fact, he was a lot of a maths geek. His other main responsibility in the Abbey was looking after the money and doing all the housekeeping and all the mathematical housekeeping, the accounts and so on. That's why he ended up as the abbot eventually. Now, his conclusion was that the tall allele must be stronger or dominant to the short allele. He called them dominant and recessive because the dominant ones seemed to be stronger than the dwarf ones. He tried to publish this research. He wanted to try and present it at his local university, but he was pretty much laughed out of the room. In fact, almost literally laughed out of the room. And as a result, even though he was doing his work in the 1840s, the research just was never actually touched again or even published until it was found again in the 1930s when the Abbey was being redeveloped and redecorated. And they found all his ledgers and all his work and all his notes in a safe that had laid undisturbed since he died in, 1884? All these conclusions that he came up with are not just relevant to pea plants. It applies to everything. In fact, he was working at the same time that Darwin and Wallace were coming up with all the background and all the observations about evolution. So really, what you had was Darwin and Wallace were doing all the observational part of the science and showing what happens and how it happens. Mendel was actually starting to work out the mechanics of what's going on almost at a molecular level, even before anybody actually knew that genes existed. Right. So according to Mendel, each characteristic has two forms. One of those forms is dominant, and you give that a capital letter by convention, and one of them is recessive, so you give that a lowercase letter. Simple enough, all makes sense. Dominant always overrides the recessive. So if the dominant is there in the pair, that is the characteristic version that will express. So for example, if the dominant is red, for like red flowers, and the recessive is yellow, as soon as the dominant is in that pair, the flowers will be red. It can be just one dominant, it could be both, it could be co-dominant, homozygous dominant, that flower will be red. Or in fact, there we go. If you have just dominant or just recessive alleles, your organism is homozygous. So capital R, small r, etc. That means homozygous. So homozygous dominant would be capital R, homozygous recessive would be lowercase r. And I'm just using R as an example. It can be any letter you want, really. If your organism's got one of each, and by convention you put the capital one first, it's heterozygous. So hetero meaning different. Capital R, small r. See? That will be heterozygous. Now, that's some of your definitions done. We've talked about alleles, we've talked about dominant, we've talked about recessive, homozygous, and heterozygous. So how does this all work then? So in practice, in real life, actually as bits of molecular biology, how does this work? Well, it turns out, as we know from a few weeks back, characteristics are passed on chromosomes that you inherit from your parents. Of a pair of chromosomes, you get one from your mum and one from your dad. And since these chromosomes are pairs, they have the same genes in the same places. There's an example. So if we say the petal for gene colour on one chromosome is up near the top, on its pair, the chromosome for petal colour will be in the same place. That makes sense, doesn't it? But it is possible that each chromosome may have a different version of it, so a different allele. For example, this one could be capital R, that one could be small r, so it could be heterozygous, or they could be the same, capital R, or small r, small r. So in this case, that will be capital R, if we're saying the red's the dominant one, and that one will be small r, if we're saying that it's the recessive. So this particular pair of the genotype will be capital R, small r, and its phenotype will be red, if red is indeed the dominant one. So yeah, as I've already mentioned, the dominant allele is always a capital letter, every time. The recessive allele is a lowercase letter, so a small letter. So there you go. And the allele pair, as I mentioned, is called, for each characteristic, is called the genotype. So the genotype for this would be capital R, small r. Homozygous genotypes would be capital R, or small r. Yeah. Okay. Now then, this is where we get into the bit where you are scoring marks on the exam. Because questions about genetics and doing crosses come up with startling regularity. One of the crosses you may well be asked to do is crossing homozygous parents. That's usually the first one of two, if they're going to go the whole hog on the genetics questions. So what you do, you pick a letter, one might be picked for you, so we'll use these. Red is the capital R and the dominant, yellow is the small r and the recessive. So the phenotypes, there you go. So the appearance, that's the dominant one, that's the recessive one. So the genotypes are there. That one, we're assuming, in fact, we'll be told that that one is a homozygous dominant. So that, in this case, will be capital R. This one is a homozygous recessive. So small r. First thing you do, work out the gametes. You work out the gametes by splitting the genotype in half, like that. And then you line them up and you Punnett square. Your Punnett square looks like that. So you put one pair of gametes down the side, one pair of gametes at the top, and then you pull one down from here to put it in there, one from there to put it in there, and so on. Now, for what is possibly going to be the second mark, I have to fill in those in, or it could be from working out the gametes, actually. What would be phenotype of those flowers if you're looking at a cross between a homozygous dominant and a homozygous recessive? Let's find out, shall we? So the F1, so first filial generation, F1, genotypes are like that. Capital R, small r, all the way across. Worked out as we did using the Punnett square. So that means the phenotypes, the appearance, are red, and red. Now remember, this is all probability. For this particular cross, there is a 4 in 4 or a 100% chance that those flowers will come out as red. So any new flower grown using that particular genotype, it's going to be red from that particular gene cross. So two homozygous parents, one dominant, one recessive, you always end up with a heterozygous genotype, which means the dominant is what's expressed. Okay? Now then, F2 generation. This is where you get two heterozygous parents, and you cross those. So you work them out in the same sort of way. Genotype is capital R, small r for both of them. You work out the gametes like that. And I would recommend you laying it out like this. Learn this layout, because you may be asked about flowers, you may be asked about peppered moths, or any one of a number of things. In fact, those tend to be the favourites, actually. Peppered moths and flowers. You lay out your Punnett square, you put your gametes in, and then you do your results. Now then, what colours are these flowers going to be? So what is the phenotype of these flowers? So your genotypes of the offspring, so the F2 generation, are capital R, capital R, capital R, small r, capital R, small r, and small r, small r. So what are the phenotypes going to be? Capital R, red. Because, as you can see, two dominants. Both the alleles are showing the dominant gene, or the dominant version, sorry. So this one, we've got a dominant and a recessive. So what colour? Red. Same again with this one, so red. Now with this one, we've got a recessive and a recessive. So what was the colour for the, if you remember, think back, what was the colour for the recessive character? Yellow. So the homozygous recessive was yellow. So this is what you get. You always get this 3-to-1 ratio. And this, again, is a common thing when it comes to the exam. They ask about this quite a lot, showing this 3-to-1 ratio. So when you've got two heterozygous parents, what you always get is a 3-to-1 ratio in the phenotypes. 3 showing the dominant, 1 showing the recessive. And what you always get in terms of the results is a quarter of them are homozygous dominant, half of them are heterozygous, and a quarter of them are homozygous recessive. 3-to-1 ratio. Now then, other possible examples that might get used and you might be asked about. Now, one condition carried as a recessive gene is cystic fibrosis. Cystic fibrosis affects mostly the lungs and digestive system, because what it does is it changes the makeup of the mucus that is produced by the lungs and the digestive system to keep everything moist, so it works properly. It does that by making the mucus thicker, and this thick, sticky mucus blocks things, blocks passages. It stops nutrient molecules dissolving into the bloodstream through the wall of the small intestine. It stops the oxygen molecules getting into the bloodstream through the alveoli, etc. Now, it can be treated. One treatment, as it shows there, includes, well, they say special inhalants. So one of these special inhalants is basically gene therapy. So in that inhaler, the patient will have a vector which is built with corrected copies of the genes which can get into the cells down the trachea, for instance, and it will thin the mucus. Because then, for while those cells are alive, because you know your cells are replaced every so often, they will be producing a much thinner normal mucus. Lung transplants are also an option for very severe cases. But as it says there, and as I mentioned earlier, another problem with cystic fibrosis is that the mucus in the digestive system is much thicker, which means there are problems in absorbing and digesting food. So in that case, a cystic fibrosis sufferer will end up taking pills that will break down the mucus to allow them to absorb the food they've taken in. Now, their life expectancies are getting longer, but it's considering that they've got all this mucus building up in the lungs and in the digestive system all the time, that leaves them prone to infections. So they don't tend to live as long. And the fact that it's on a recessive gene means it's a lot harder to eliminate from the population, because people can be carriers. They can carry it as being heterozygous without knowing that they've actually got it. Because remember, all these bits of Mendelian genetics, all of those Punnett squares, all that means is a chance of. It's a 1 in 4 chance with 2 heterozygous parents for 2 carriers. It's a 1 in 4 chance of making a baby that has cystic fibrosis. It's a 1 in 4 chance. That doesn't mean every 4th one is definitely going to be cystic fibrosis. Just that they could have 4 kids, all of them with cystic fibrosis. They could have 12 kids, none of them with it. It's all probability. So how is it inherited? Well, I've already mentioned that. CF is caused by a recessive allele. So that means that if you have cystic fibrosis, you've got a double dose of the recessive, one from your mum, one from your dad. Sickle cell anemia, in terms of its genetics, is very similar, except it's obviously a very different disease because sickle cell anemia creates malformed hemoglobin. The sickle cell hemoglobin creates that characteristic sickle shape, which is also more brittle. So that means even the working red blood cells, the hemoglobin isn't as tough and flexible as it's meant to be. Because normal red blood cells, they can move freely through blood vessels. They can bend and flex and fit through small gaps just by being, bent. With the sickle cell red blood cells being stiffer and more rigid, they can get stuck and they can tear blood vessels, causing internal bleeding and so on. People with sickle cell tend to be in pain a lot of the time. You might know some. Again, it's caused by a recessive allele. So that means they've got the small s. The strange thing is that if you are a carrier of sickle cell anemia, so if you are heterozygous in that particular allele, you are actually immune to malaria, believe it or not, because if you are a carrier, so if you are heterozygous for sickle cell anemia, if a plasmodium parasite, which is the cause of malaria, gets into your bloodstream and it attacks red blood cells to feed off them, which is what it does, your red blood cells, if they have the heterozygous trait, they are resistant to the plasmodium parasite. They will literally fold up so the parasite can't get a grip on the red blood cell. Thalassemia works in the same kind of way. There is something about it that stops malaria parasite feeding off the red blood cells. So really, sickle cell anemia and thalassemia are both defenses against malaria. that our species has evolved